The overall goal of the following experiment is to obtain statistical maps comparing the difference of resting state fMRI scans between healthy controls and patients with epilepsy. This is achieved by obtaining fMRI data in subjects with temporal lobe epilepsy as well as healthy control subjects. As a second step, the fMRI is pre-processed, which prepares the data to be statistically analyzed. Next, the pre-processed fMRI data is analyzed in order to obtain statistically valid comparisons between the two groups. Results are obtained that show differences in brain networks among patients with epilepsy compared to brain networks of healthy control subjects, based on statistical differences in brain network maps between these two groups.
It's become increasingly apparent that even epilepsies that are manifest by focal seizures are emergent properties of diffuse network disturbances.
The idea for this technique came after recognizing the importance of the default network during seizures and wondering whether the default network is abnormal between seizures in temporal lobe epilepsy.
The study population for this protocol should include 3 groups: right temporal lobe epilepsy patients, left temporal lobe epilepsy patients, as well as healthy controls. A total of about 35 subjects are recommended. The epilepsy subject groups should be patients who are diagnosed to have temporal lobe epilepsy, such as candidates for anterior temporal lobe resection as determined by video EEG monitoring, PET imaging, and neuropsychological testing. Assure that all subjects have normal brain MRIs and